Good morning students. Today I am going to explain about plant tissue culture part 1. This is very important lesson and this is very small lesson. Okay. So this is very small lesson as I told you. So in this lesson you are going to learn about the techniques of the biotechnology. Okay. So you are going to learn the techniques of the biotechnology. Introduction, I am trying to say that is growing plants, that is if you are taking the plant cell, that is the without the cell wall of the plant cell is called protoplast, right? So that protoplast cells, tissues and organs away from their natural or the normal environment under the artificial condition. The cell is usually growing under the normal condition, that is naturally it is growing as a big cell one, right? So the normal cell become a normal cell is undergoes the growth, reproduction, cell division, everything is takes place, it is takes place in the normal condition. But all those things like a growth of the cell, division of the cell division, everything is takes place in the under the artificial condition. That condition is made by us. Okay, so that is called tissue culture. In vitro means that is outside the body, that is outside the plant body we are growing the plant, and without the body of the plant is called in vitro. In vivo means inside the plant cell is growing and multiplying is called in vivo. So this is the meaning of in vitro. That is in Latin word, it meaning is the plant growth is takes place in the glass or the test tube. The single explant can be multiplied into several thousands of plant in short time of period space under the controlled condition. We are giving a controlled conditions like particular optimum temperature, pH for the growth of the plant in the test tube or the container it is growing. So that it is called single explant means small twig of plant or the single cell or the tissue of the plant is used for the multiplication of the plant under the artificial condition. That single tissue or the twig or the cell is called explant. So PTC meaning is plant tissue culture. So by using of tissue culture technique we are growing the multiply the plants as well as it is used for the plant research, and it is also indispensable tool for the regeneration of transgenic plants. Okay, very important and economically important plant can grow by this method. Apart from this, you can grow elite varieties, very rare varieties of plants which is very rare in the world, that elite varieties, and conservation of endangered places. Conservation of endangered plants means that is if the plant is growing under the, that if our world having only 10 trees of the apple is there, so the tree 10 apple tree is going to be destroy, or it is going to it is going to exceed from our world. So that we have to conserve or protect that particular plants by using this PTC technique. And also we can make production of virus free plant, that is germ free plant we can grow, and germ plasm, that is the DNA and seed of the plant also preserve by using this method. And also industrial production of secondary metabolites like antibiotics, vaccines also by using this method we can prepare. And father of the plant tissue culture is Gottlieb Haberland, is the German botanist, proposed the concept of totipotency. That you know that totipotency and totipotent you have studied in the first lesson itself. Okay, that is he is a person, the first person to culture the plant cell in artificial condition using the mesopyle cells of Lamium purpureum in the culture medium and obtain a cell proliferation. He is regarded that father of tissue culture. So Laminum purpureum is a plant. In that plant he have taken mesophyll means only one leaf cell he have taken. So that particular cell is divided into hundreds of cells is called the this thing, that is a so plant tissue culture means, that is suppose this is an example, this is explant. It may consisting of group of cells or the tissues or the small tissue of the carrot plant is taken, and it is kept in the medium that is consisting of nutrient materials which is necessary for the growth of the plant, and which is the material or the composition which is present in the medium, it's also present in the soil. So next is it is growing as an callus. Callus means undifferentiated mass of the growth of the plant. So from the callus we will be getting a early embryo, then we'll be getting a late embryo, then we'll be getting a plantlet. Okay, this plantlet is become a plant. Okay, so this process, that is from the single cell we'll be getting a whole plant, that is called a plantlet. Small plant is called totipotency. The DNA or the cell is can able to give the full plant is called totipotency. Next, basic concepts of the tissue culture you should know. So leaf you can take, leaf small piece, that is small tissue, that is group of leaf cells are there in the leaf tissue, is taken and kept in the petri plate. Then it is undifferentiated callus mass. In this it is undifferentiated into the callus mass. Okay, so this callus mass, the callus is separated and single cell is cultured. Okay, so what happened, undifferentiated callus is produced and these is separated into a single cell and culture, and further it is growing as in the test tube. So totipotency means the genetic potential when cultured in the nutrient medium. Nutrient medium means nothing but its temperature, particular pH and particular nutrients which is very very necessary for the growth of the plant will give rise to the complete individual plant from a genetic potential. The single gene will give you the full plant by their potential. That potentiality of the gene is called totipotency. Next, differentiation, the process of biochemical and structural changes, that is the cell which is growing in the artificial nutrient medium which is undergoing some biochemical structural changes so that it is differentiated into from one cell to differentiated as a tissue. So that is called differentiation. What changes? That is biochemical and structural changes. Redifferentiation means the further differentiated plant part is again it is differentiated cells into another type of cells. Example, when the competent cells of the callus have the ability to form a whole plant in the nutrient medium, this phenomenon is called redifferentiation. From the differentiation, the one type of cells get differentiated. This differentiated cell will give you the another type of the cells is called redifferentiation. Dedifferentiation means this is just a reversion of mature cells to a meristematic cells. Mature cells is become a meristematic cells leading to the formation of callus is called dedifferentiation. This redifferentiation and dedifferentiation is an inherent capacities, that is the plant itself having some inherent character or the inherent capacities to undergoing a redifferentiation and dedifferentiation. Not only in the plant tissue culture, the plant which is normally growing in the land will have the tendency phenomena of the redifferentiation and dedifferentiation. Next, the plant tissue culture is described, that is in vitro aseptic growth of the any plant part. That is plant tissue culture na ஒரு பிளான்ட்டு இன்விட்ரோவா, அதாவது ஒரு டெஸ்ட் டியூப்லையோ ஒரு கிளாஸ் கண்டெய்னர்லையோ ஒரு பர்டிகுலர் டெம்பரேச்சரில் ஒரு பர்டிகுலர் பிஹெச்சில் எந்த ஒரு ஏசெப்டிக் கண்டிஷனானது ஸ்டெர்லைஸ்டாக, எந்த பாக்டீரியா வைரஸ் எதுவுமே க்ரோ ஆகாமல் ரொம்ப ஸ்டெர்லைஸ்டாக ரொம்ப க்ளீனாக நம்ம க்ரோ பண்ணி கொண்டு வரதான் பிளான்ட் டிஷ்யூ கல்ச்சர். அண்ட் தீஸ் பிளான்ட் பார்ட் ஆர் த எக்ஸ்பிளான்ட் பார்ட் மஸ்ட் பி செலக்டட் ஐசோலேட்டட் ஃப்ரம் த ரெஸ்ட் ஆஃப் த பாடி. ஒரு பிளான்ட்டோட லீஃப் வேணுமா ஸ்டெம் எடுக்கணுமாங்கிறத நம்ம தான் கரெக்டாக பார்த்து செலக்ட் பண்ணணும். த எக்ஸ்பிளான் மஸ்ட் பி எக்ஸ்பிளான்னா டிஷ்யூ. Plant tissue must be maintained controlled by physically and chemically defined condition. ஸோ அதை வந்து பர்டிகுலர் டெம்பரேச்சர், பர்டிகுலர் பிஹெச்சில், பர்டிகுலர் கெமிக்கல், என்ன கெமிக்கல் உள்ளே இருக்குது, என்ன நியூட்ரியன்ட் கொடுக்க போகிறோம் அப்படின்ற தெரிஞ்ச நியூட்ரியன்ட், என்னென்ன நியூட்ரியன்ஸ் இருக்குது அந்த மீடியமில், ஸோ பிளான்ட் எப்படி வளரும், ஸோ அதுக்குள்ளே என்னென்ன கெமிக்கல் நியூட்ரியன்ஸ், கார்போஹைட்ரேட் இருக்கா, விட்டமின்ஸ் இருக்கா, மினரல்ஸ் இருக்கா, ப்ளஸ் அதோட பர்டிகுலர் ஆப்டிமம் டெம்பரேச்சர், ஆப்டிமம் பிஹெச் எல்லாம் கொடுக்க முடியுதான்றதை செக் பண்ணணும். நெக்ஸ்ட், ஏசெப்டிக் கண்டிஷன் மஸ்ட் பி மெயின்டைன். Aseptic means sterilized condition or the very clean condition or the very hygienic condition should be maintained, which requires the high degree of cleanliness in the laboratory. Laboratoryல plant வளரும்போது கண்டிப்பாக அது வந்து ரொம்ப க்ளீனாக இருக்கணும், இல்லைனா பூச்சி, ஃபங்கஸ் இதெல்லாம் ஈஸியாக க்ரோ ஆயிடும். Explant means the tissue which is taken from the selected plant transferred to a culture medium, often to establish the new plant. This is just definition of explant. Next, explant same thing, and laboratory facilities for PTC. Washing facility for a glass vase and oven. So if you are doing the plant tissue culture in the laboratory should need these following characters like washing facility. Always the tap with the water connection to wash the glass vase, and also ovens for the drying the glass vase. Okay, ovens means hotter oven. It should dry our glass vase. If we are cleaning the glass vase, then you should keep in the hotter oven that should remove the all water or the moisture or the wet in the glassware. So that could be we can use for the plant tissue culture, so that we can maintain the aseptic condition. And also the medium preparation and the room with the autoclave electronic balance pH meter is there. So if you are going to prepare the medium, that is nutrient medium, for that how much gram you are going to take, how much water you are going to take, what is the pH and what is the electronic balance. So and these are all the things you should need. Okay, so what is the meaning of this point, I'll explain you in the after coming slides. Okay, so the transfer the area of a sterile room with a laminar airflow and bench with a high efficiency particulate air filter to maintain the aseptic condition. There is one instrument is there, its name is high efficiency particulate air, that is laminar airflow is like a table, like table mari UV light oda irukka or apparatus or instrument. In that instrument only we will be using our medium, nutrient medium, will be pouring or will be dispensing the medium, our prepared nutrient medium, into the test tube or any glass container that should be sterilized with the water and a hotter oven. So that instrument, table like instrument, is called laminar airflow. So culture facility, growing the explant inoculator, that is which type of explant you are going to inject or you are going to insert or you are going to inoculate on the culture tubes at 22 to 28 degrees centigrade. And elimination of light, that is light also needed for the growth of the plant because it always absorb the sunlight. So that the elimination of light is 2400 lux. Okay, where a photo period is 8 to 16 hours, it should be in the light, and relative humidity should be a 60%. If you are growing the plant under the artificial condition, you should need a relative humidity about a 60%. The photo period, it should be keep in the light of the 2400 lux with the time of 8 to 16 hours. That's all. So techniques involved in the plant tissue culture means the sterilization to get rid of microbes such as bacteria, fungi in the culture medium or in the vessels or in the expanse. Okay, culture medium means what medium you are going to prepare, nutrient medium or Murad scoop medium, different kinds of medium is there. That is a enriched medium, or the medium will help the growth of the bacteria, growth of the plant, and which type of vessels like test tubes and container, and which type of cells you are going to use. So these are all the things is main important for the this thing to maintain the sterilization of medium. So maintenance of aseptic environment, how can you maintain the aseptic environment during in vitro the tissue culture, the sterilization of glasswares, forceps, scalpels, and also all accessories in the wet stream sterilization. Okay, so you should use the sterilization technique. Two types is there. You should use the hotter oven is a dry heat sterilization technique, and moist heat sterilization technique is autoclave. Under the dry heat sterilization technique, you should keep your all test tubes, containers, forceps, scalpels, whatever you are going to use for the plant tissue culture technique, all should be washed with the water, and also you should be wrapped with a newspaper and should keep inside the hotter oven for 1 hour. After a 1 hour, this all the wet and all moist of the all your glassware, forceps and scalpel will be removed, dried, and it should be rid of all microbes or the bacteria, fungi, and you will be getting a fresh glassware, forceps, scalpel without any bacteria and fungal growth. Okay, that is about the dry heat sterilization. If you are coming to the moist heat sterilization, you should use the autoclaving the medium. You should using the autoclaving the medium. Autoclaving means, that is autoclave is like a cooker. Your house you will be using a pressure cooker. Your house you will be using a pressure cooker. In that pressure cooker we'll be using the will be taking a water and will be keeping our medium, our weighed medium, our nutrient medium powder is added in the test tube, and water is added and it is mixed, and you should be keep in this autoclave medium. So we should keep in the medium. Okay, so that's all about your autoclaving. That autoclaving temperature is 121 degrees centigrade for 15 to 30 degrees centigrade, or else you can dip in the 70% of ethanol or by following the flaming or the cooling. Flaming means in the Bunsen burner you should show the mouth of the conical flask, and also you should wait for the cooling of that. So sterilization of culture room. So where you are going to add this explant into the culture medium, in that culture room only. So floors and walls are washed with the first with the detergent soap water. Then with the 2% of the sodium hypochlorite or the 95% of ethanol is used to clean the room. And laminar airflow, as I said earlier, that is used to sterilize by clearing the work surface that is inside the laminar airflow by using the 95% of ethanol and exposure to UV radiation for 15 minutes. UV light and normal illumination light will be there fixed in the top of the laminar airflow. Once you clean that laminar airflow table with the 95% of the ethanol, after that you switch on the UV light and close that room and come back to the normal area. After a 15 minutes you go inside, switch off the UV light, then you can start do your work. UV light will kill the all microorganism inside the laminar airflow. Also you are wiping with the 95% of the ethanol, because this laminar airflow you will be using as the working surface to add the medium into the culture plate or in the test tube, and where only the explant will grow as the new plant. So sterilization of nutrient medium. The culture media are dispensed in the glass containers and plugged with the non-absorbent cotton. So once your medium is added in the test tube, then you are putting the cotton to as make it as an லிட். ஓகே, லிட்டு நீ போட முடியாது இல்லையா. ஒரு நான் அப்சார்பன்ட் காட்டன் எடுத்து சுருட்டி அது மேலே வச்சு சீல் பண்ணிட்டா இட் இஸ் அன் குட். ஆனால் ஆல்சோ யூ ஷுட் கவர் திஸ் ஃபுல் செட் ஆஃப் ஒரு 10 டெஸ்ட் டியூப்பில் நியூட்ரியன் மீடியம் மூடுறேன்னா எல்லாத்துக்குமே நான் அப்சார்பன்ட் காட்டன் போட்டுட்டு அதை வந்து ஒரு பீக்கரில் வச்சுட்டு அது மேலே ஒரு நியூஸ் பேப்பர் போட்டுட்டு யூ ஷுட் கவர் அண்ட் கீப் இட் ஃபார் த ஆட்டோக்ளேவிங் 121 degree centigrade for 15 pounds. That is 15 pounds mean measurement of the pressure for a 15 to 30 degrees centigrade. The plant extract vitamins, amino acid and hormones are sterilized by passing through the suppose vitamins amino acid லாம் நீ வந்து clear பண்ணணும்னு நினைக்கிற ஸ்டெரிலைஸ் பண்ணால், if you are keeping this in the autoclave they will be sterilized. So it will be sterilized and it is passing through the millipore filters with 0.2 millimeter and pore diameter, then added to the sterilized culture medium inside the laminar airflow chamber under the sterile condition. The sterilization of explants. The plant material to be used for a tissue culture should be surface sterilized by exposing the material in the running tap water. So running tap water la and the explant culture wash பண்ணும், then treating them in the agents like 0.1% of mercury chloride or the 70% of the ethanol under the aseptic condition inside the laminar airflow chamber. That is our working surface area. So this is about the sterilization is over. Techniques that is involved in the PTC is first one is sterilization, maintenance of the aseptic conditions, and second one is media preparation. So the media how you can prepare. The plan is growth regulators and culture conditions such as temperature, pH, light, humidity. As per the principle of tissue culture, you should the MS medium, that is MS nutrient medium is Murad's Skoog. Murad and Skoog is a person. They discovered this nutrient medium for the growth of the plant under the artificial condition in the year of 1962. It is commonly used because it consisting of carbon sources, suitable vitamins and hormones which is necessary for the growth of the plant. And also this Murad Skoog medium or B5 medium by Gamborg is a scientist in the year of 1968. White medium is discovered by White scientist in the year of 1943. NITS medium is a NITS in the year of 1969. And a medium may be a solid or semi-solid or the liquid. That is for the solidification we'll be adding gelling agent like a agar. Agar is nothing but it is taken from the complex mucilaginous polysaccharides obtained from the marine algae or the seaweeds used as a solidifying agents in the media preparation. Seaweeds means unwanted plants which is growing in the seas. That seaweeds is taken and it's have the tendency to give the solidifying the agent. So in that solidifying agent we'll be getting in the powder form in the name of agar-agar. That agar-agar is added in the this NITS medium or the Murad Skoog medium or the white medium or the B5 medium. All this kind of medium we can add. By adding of this medium, the medium is become a solid, or by adding of very little bit amount we'll be getting a medium in the semi-solid state. By adding a correct amount we'll be getting a solid. Without adding of this agar-agar in the medium we'll be getting a liquid. Okay, so you have taken skippies or the what is this, jellies you have taken. Okay, you have you may ate, right? So it is somewhat in the semi solid state, that is a semi solid and somewhat a solid state is given by this agar agar. Next, this is does not need for you. And culture condition, the culture, if you are growing a plant under the plant tissue culture under the artificial condition, you should maintain the pH for a 5.2 to 6 pH. Okay, that is acidic pH you have to maintain for the growth of the plant. And also the medium we have prepared, that is Murad's Cube medium, is present with many carbon sources and many vitamins is there. Temperature culture should be incubated normally at constant temperature of 25 degrees centigrade plus or minus 2 degrees centigrade for optimal growth. Humidity and light intensity. Humidity means the culture it requires a 50 to 60 degrees centigrade percentage of relative humidity for 16 hours, and its photo period, that is illumination, the light period for this, that white fluorescent tubes approximately 1000 lux. Okay, 1000 lux of the fluorescent white light. Okay, fluorescent white light they will be using up to the 1000 lux for a 16 hours we have to give for the plant. And also it need a humidity, that is moist thing, that is 50 to 60 degrees centigrade. An aeration to the culture can be provided by shaking the how can you give the aeration? By shaking the flask or the conical flask or the tubes, the liquid culture medium in the automatic shaker. Automatic shaker is an instrument. By keeping this test tube or the conical flask, we can switch on the switch of the automatic shaker. It will shake for a particular period, so that we will give you can generate the aeration or the oxygen to the plant. Humidity you should maintain for 50 to 60 degrees centigrade. So by doing this kind of the culture condition, the induction of the callus, that is callus means that is undifferentiated mass of the plant. Okay, so the explant of the 1 to 2 centimeter sterile segment is selected from the leaf or the stem or the tuber or the root of the plant, and it is transferred to the Murad's Cube nutrient medium supplemented with the auxins and incubated at 25 degrees centigrade, which may be a plus or minus 2 degrees centigrade in an alternate light, and dark period of 12 hours to induce the cell division. Soon the upper surface of the explant develops into callus. Okay, so the supplemented with auxins, the Murad's Cube medium abdina, that should be as supplemented with the auxins. Auxins is a hormone which is necessary for the growth of the plant, and it should be incubated means its growing condition, growing setting temperature இதுதான். Okay, so callus mass is unorganized growth of plant cells or the tissues in the in vitro culture. So this is just diagram of the culture. So embryogenesis means the callus which cells undergoes a differentiation produce the somatic embryos known as embryoids. It produces somatic embryos means it produces the embryos of the roots and leaves and stems. The embryos are subcultured to produce as the plantlets. If it is growing in the test tube, you should be take and keep it in the some other else. Yeah. Hardening period. Hardening period means hardening is a gradual exposure of the in vitro developed plantlets in the humid chambers in the diffused light for acclimatization. So hardening, once the plantlets are grown in the test tube, you can take and keep it on the field work. Okay, normal grow. You can grow under the normal field condition called hardening. So types of plant tissue culture: organ culture, meristem, protoplast and cell culture. Organ culture means the culture of the embryos, anthers, ovaries, roots, shoots and other organs can be grown by this method. And meristem culture is the tip of the tissue will be using. The older leaves are removed from the culturing and the carnation. Then magnified view of shoot tips after the removal of larger leaf. Shoot tip with growing tip after removal of enclosing the leaves. Next, protoplast culture is the protoplast means the plant cell without the cell wall is called protoplast. It is possible to regenerate the whole plants from the protoplast cells. First, you have to do isolation of protoplast, next fusion of protoplast, next culture of protoplast, then selection of somatic hybrid cells. Isolation of the small bits of plant tissues like leaf tissue are used for the isolation of plant protoplast. The leaf tissue is immeasured in 0.5% of macrozyme and 2% of Onotsuka cellulase enzyme and dissolved in 13% of sorbitol or the mannitol at pH 5.4. It is then incubated overnight at 25 degrees centigrade. After a gentle teasing of the cell, the protoplasts are obtained, and these are then transferred to 20% of sucrose solution to retain their viability. They are then centrifuged to get pure protoplast as differentiated from a debris of cell walls. So fusion of protoplast is done through the use of suitable fusogen like a polyethylene glycol is used to paste their two protoplasts. The isolated protoplasts are incubated at 25 to 30% of concentration of the polyethylene glycol with the calcium 2 plus ion, so that the protoplast shows the agglutination, that is clumping of the different protoplasts, and fusion takes place. And culture of the protoplast, the Muratskuk liquid medium is used with some modification in the droplet plating and microdrop array techniques. The protoplast viability tested with the fluorescence diacetate before the culture. The cultures are incubated in the continuous light 1000 to 2000 lux at 25 degrees centigrade. The cell wall formation occurs within the 24 to 48 hours, and the first division of new cells occurs 2 to 7 days of culture. And selection of somatic hybrids, the fusion of product of the protoplast without nucleus of the different cells is called as hybrids. So the following the nuclear fusion is happened, this is called somatic hybridization. The fusion product of protoplast without the nucleus is different of different cells is called hybrid. Nucleus fuse ஆகாம மற்றதெல்லாம் fuse ஆச்சுன்னா hybrid. nucleus தான் fuse ஆச்சுன்னா that is somatic hybrids. This is just diagram of protoplast, and production of secondary metabolites are used for this. This PTC is used for the production of secondary metabolites like alkaloids, flavonoids, terpenoids, phenolic compounds, recombinant proteins, and also it is used for the production of byproducts of the cell metabolism. If the cell is undergoing any glycolysis or glyconeogenesis process, it doing some byproducts, it leaves some byproducts is used as in secondary metabolites, which is necessary for the growth of humans, as like vaccines and antibiotics. Biosynthesis of and isolation of indole alkaloids from Catranthus rosas plant cell culture. The process of the secondary metabolites can be scaled up auto-producing the bioreactor for the commercial production, and biotransformation, elicitation, immobilization have been used to make the cell suspension culture more efficient in the production of secondary metabolites. Cell suspension culture is prepared by transferring a portion of callus into the liquid medium and agitated using a rotatory shaker, that is medium shaker. The cells are separated from the callus tissue, and it is used for the cell suspension culture. These are all the table that is produced by plant sources, which is giving a different types of secondary metabolites, like a digoxin is given by the digitalis purpura, that is used for the cardiac tonic, that is in the heart patients. And codeine is papaver somniferum is using analgesic, and capsaicin capsicum annum is producing rheumatic pain treatment. We can use this. Capsaicin is produced from the capsicum, and vincristine is produced from the Catranthus rosas, anticarcinogenic. Quinine is given by Cinchona officinalis, anti-malarial drug. Against malaria, we can give the quinine drug, which is taken from the Cinchona officinalis. So that's all about your plant tissue culture. I hope you all understand this. Thank you.